as mentioned, my name is Parth Sitlani, and I'm the Associate Director of Commercial for Invivoscribe. Today, we'll be talking about immunoglobulin and TCR testing in heme malignancies. We'll start with a little bit of the history of this testing, how these genetic rearrangements happen, how they've been tested in the past, how they are tested now in most laboratories, and then how they will likely be tested in the future. So let's get started talking about gene rearrangements, how they happen, and the clonality testing process. So most human cells, when we consider the differences between our lung cells and our brain cells and our skin cells, we anticipate that the DNA of those cells will be identical between a given individual. But immunoglobulin and T cell receptor genes have this process of rearranging within lymphocytes specifically as a part of the adaptive immune system of the body. So at these genetic loci, we have a rearrangement process that occurs during cell development that causes these gene segments that are very similar to each other in sequence, but slightly different to rearrange. So a portion of that DNA is cut out and pasted back together. And this selection of which VDJ gene is pasted together happens somewhat randomly, leading to a VDJ gene rearrangement that is relatively unique because this rearrangement is happening semi-randomly across each of these lymphoid cells, leading to a slightly different selection of VD and J. Now, if we were to take a look at our lymphocytes, what we would find is, in theory, each of them should have a different immunoglobulin, if it's a B cell or T cell receptor gene locus as it develops. And when we look at the actual VDJ rearrangement, for example, here, if this is B cell X, it has DNA sequence at IGH of V1, D1, J1, and that cutting and pasting process you can see here in red is also slightly different. This DNA sequence of the bases is an actual fingerprint that can be connected directly to B cell X. And the same can be said for B cell Y, where this DNA sequence with its different V gene, different D gene, different J gene, and difference in length, that is a fingerprint now directly connecting to B cell Y. And again, B cell Z, we have a different selection of genes and length, and even the mutations and sequences within these rearrangements. Those are now a fingerprint that can be connected back to B cell Z here. Now, in a polyclonal cell population, this is a healthy immune system. What we expect is that we should be able to go ahead and count these fingerprints. And if we were to take a look at this polyclonal cell population here, what we'll find is that B cell X's fingerprint can be found one time, B cell Y's fingerprint can be found one time, and B cell Z's fingerprint can be found one time. However, in a clonal cell population, if we go through and count these rearrangements, what we find is that B cell X is represented once, B cell Y is represented 12 times, and B cell Z is only represented once. Now, this can, in theory, be a part of a healthy immune system responding to infection or vaccination. But in the case of hematologic malignancies, what we're really concerned about is that these lymphoid cells are malignant. And in that case, we can use this result here, the overabundance of B cell Y, to confirm the presence of that malignancy. But how do we go about counting fingerprints? They are, of course, just animations here, and they are cells in this section over here to the left. And that's when we get into the history of clonality testing. So clonality testing has been done in the past using Southern blot. I won't be talking too much about Southern blot, but in more modern labs, what we're seeing most often is the heteroduplex analysis with gel detection. We're also seeing fragment analysis with capillary electrophoresis machines, and we're seeing next generation sequencing where we really start to count our actual fingerprints. So let's look at each of those detection methodologies. Here on the left, we have gel detection. where you can see generally a polyacrylamide, sometimes an agarose gel, being used to show the difference between a positive, a negative, and somewhere in between, where that interpretation is a very visual process to take a look at a picture of a gel and call a result to confirm the presence of a malignancy. And this is done using heteroduplex analysis. Capillary electrophoresis uses fragment analysis. If you recall, I mentioned earlier that the length of each of those rearrangements is slightly different. And in a polyclonal cell population, we expect that distribution of lengths to be normally or bell-shaped distributed at a given average length. However, if B cell Y becomes overabundant, the length the peak representing the length of B cell Y should be higher and stand out from that normal bell-shaped curve. And you can see that here, this peak is significantly higher than what is in the background. And you can see the negative background is significantly different as well. Now, with next generation sequencing, we're actually doing a real counting. We count the reads that we identify, but we don't look at a band on a picture. We don't look at peaks on an electrophoresis graph. Instead, what we look at for next generation sequencing is the actual DNA sequence of that VDJ rearrangement. So Invivoscribe has assays for all three of these methodologies, and we try to harmonize them across the three different platforms. So most assays do follow a very similar procedure of DNA extraction, single-step PCR setup, and then detection using that specific method. So here's heteroduplex analysis, here's capillary electrophoresis, and here is next-generation sequencing. Now, each of those methodologies has its benefit and its pitfalls, and we'll discuss those depending on where your lab is located, what you have access to, and what the needs of the hematologist are. So starting with heteroduplex analysis, what we do is we take our PCR amplicons that have all of our VDJ rearrangements amplified, and we heat denature them at 95 degrees for 5 minutes, and then we snap chill them on ice. What that means is that when you have a population of clonal cells, when they are snap chilled back together, they very easily find their reverse complement partner and lead to a band when they travel through the gel. When you have a polyclonal population of cells where each of the VDJ sequences are diverse and different from one another, when they are heat denatured and snap chilled together, they will find their closest physical partner, but they may not find an exact reverse complement in their DNA strand. And what that means is that there is secondary structure, hairpins, and loops that are formed that end up causing these molecules to traverse very slowly through the gel and cause a smear or really even get stuck up at the top of the gel. So we know that within our valid size range here in blue are potentially clonal rearrangements, and up here in red are sequences that did not travel through the gel due to secondary structure. And of course, here sample in well 2 is the positive control, sample in well 3 is a 10% positive control, and the sample in well 4 is the negative control. And we can very clearly distinguish between these three different result types. So what are the benefits of this gel detection? So we can, of course, do this with just a gel electrophoresis setup in the lab. You don't need any major instrumentation. That also leads to a very low cost per sample and a very rapid turnaround time. And the interpretation is very easy. As you saw, I showed band, no band. There's not a lot of calculation or measurement that needs to go into identifying if a clonal rearrangement is present. Now, the downsides of gel detection are lower sensitivity. You saw what a 10% positive control looks like. Samples at about 5–6% become a little bit harder to detect by gel detection and can have a very, very faint band. Sizing, of course, is a potential issue. When we look at a rearrangement of 350 base pairs versus 360 base pairs, you usually cannot tell that difference between gel electrophoresis platforms. And then if we have two clonal cell populations or a single cell population that has two rearranged chromosomes, we cannot identify multiple clonal rearrangements. They'll be too close together and their band will just be one very large band. So moving on to capillary electrophoresis, I mentioned to you earlier that the size of the rearrangements are slightly different between the different B cells. And as many things in nature, when you have an average value, we actually have values that have a normal or bell-shaped curve distributed around that average. So within, let's say, this peak here, there is hundreds and hundreds, maybe thousands and thousands of different rearrangement sequences that all have the same length, but they still form this average bell-shaped curve, even with the single base pair resolution. Now, we can still see that bell-shaped curve here in the positive example on the right side of your screen. However, we also see that when one cell is overrepresented, that cell's rearrangement size, let's say it's B cell Y, if this is 325 base pairs long, then what we will find is a direct connection between this and this rearrangement size being overrepresented here when we saw that B cells Y was a very highly represented population of cells. So the benefits of capillary electrophoresis, we have much higher sensitivity and actually much better specificity than gel. It's a newer technology. The cost per sample is low on a per test basis, and the turnaround time is quite fast, but it does assume that you have the instrument in place already. So this is single base pair resolution instruments such as ABI, Seek Studio, ABI 3500, ABI 3730. We are investigating some of the other third-party instruments from brands like Promega and Qiagen. We will have some data for those hopefully in the near future. Another big benefit of capillary electrophoresis is that it's been very well standardized. Invivoscribe was involved with the Biomed 2 Euroclonality group for development of the standardized clonality detection process. And that process is now over 20 years in standardization. So there's quite a lot of literature and guidance available for showing example results and how to do the interpretation. Now, the pitfalls of electrophoresis is you do need that one base pair resolution instrument in your lab. Now, if your lab is doing microsatellite instability or other forms of Sanger sequencing, you may have access to this instrument already, but you do need to have that. Otherwise, there is some upfront cost to doing electrophoresis testing. There are many tubes in valid size ranges, which connects into the next point, which is the interpretation expertise. Since clonality testing has been standardized for 20 years, there are many experts that know how to interpret these results. There is a lot of guidance and literature available, but it does take a little bit of skill to be able to clearly differentiate when a peak is high versus when it is just a little bit of a irregular polyclonal sample. Moving into NGS. So for NGS, we have a 7-step procedure of an assay. Again, up until PCR, our assays are very similar. We then have to prepare the assays for sequencing, which means we have to purify the amplicons, remove any primer dimers and off-target amplicons, and then we quantify and make sure our library is ready to load in a way that's not going to over or under cluster our sequencer. So the actual wet lab procedure for LymphoTrack, which is our next generation sequencing product line, is highly similar. And then you have to do the longer process of actually doing the sequencing. Now, of course, the benefits of next generation sequencing are going to be extremely high sensitivity. We've seen rearrangements detected at about 2.5% clonal cell population in a background of polyclonal cells. The interpretation is very straightforward. There's not as much expertise required because it's objective and there's numbers and the counting is already done by the software. Somatic hypermutation and residual disease can all be done in the same assay at the same time when you're using next generation sequencing. Now, the pitfalls of next generation sequencing are, of course, the instrument cost to start up is quite high. The cost per test can also be a little bit higher than gel and capillary electrophoresis detection. That depends significantly on the batch size. The number of samples you can fit into a given sequencing instrument reduces the total cost per test pretty significantly when you're doing sequencing. Turnaround times are a bit longer as sequencing instruments usually take about 2 to 3 days to run. This is, of course, already being alleviated as you hear the news from Illumina, Oxford Nanopore, Ion Torrent. As their sequencers get faster and faster, we're seeing shorter and shorter turnaround times for NGS. One of the interesting pitfalls we found in more recent times is the technology is a little bit newer, so we don't have as many technicians out in the field that are comfortable with it. But of course, Invivoscribe is available to help with that process. So now to quickly get into the Invivoscribe portfolio portions, especially as it relates to the gel and capillary electrophoresis assays. So Invivoscribe has a full range of products for immunoglobulin and T-cell receptor targets. That includes the Ig heavy chain, Ig kappa chain, and lambda chain, as well as for T-cell receptor, we have beta, gamma, and delta assays available, and the translocations that are very common in heme malignancies, such as BCL1, BCL2, BCR-ABL, and PML-RARA. Similarly, IBD assays are available where they are registered for the clonality tests. Now, how do you know which test to use? These tests are designed to amplify as many possible rearrangements as can be done with a single target. Here you can see, for example, in follicular lymphoma, using the immunoglobulin heavy chain, you can detect 84% of cases that are known cases of follicular lymphoma. And with the Ig kappa chain, you can detect 84% of rearrangements. But these are not the same 84%. If we combine those targets together, then we can clearly differentiate 100% of cases using these assays together. So what that means is there are targets that can be used. We can combine IgH and IgK to get higher detection for B-cell malignancies. We can combine TCR beta and TCR gamma to get higher detection of T-cell malignancies. This does mean when you combine these together that you get higher detection. And if you do them at the same time, they all can be run side by side on the same instrument, on the same PCR plate. Your time and your labor cost is significantly reduced. Now, this can get a little bit difficult with the number of different diseases and the subdivisions of hematologic malignancies. So Invivoscribe does have a clear chart that helps to show which assays should be used for which different disease types. So here near the top, you can see we have primarily B-cell malignancies, and in dark blue are the primary targets. So you can see that follows the chain of IgH and IgK. And then down here in the T-cell diseases, you can see that the primary targets are TCR beta and TCR gamma. But the great news is that we can cover almost all B and T cell malignancies using these assays. So to summarize just this portion of the presentation, we have benefits and pitfalls of gel detection. I will quickly just show all of them together so you can see them at the same time. And the point is that the technology has evolved from gel detection to capillary electrophoresis and now to NGS, but a majority of labs actually are still right here in the electrophoresis phase because it is highly sensitive, but but still relatively low cost per sample. So it strikes a good balance to meet that newer technology as compared to gel detection without breaking the bank as far as cost per sample and turnaround times. Now, I do want to talk about the latest technology as well, because as we mentioned, we were talking a little bit about old technology, a little bit about modern testing, but also want to really focus in, spend some time to talk about our next generation sequencing assays, because this is the more cutting edge technology that's more newly available. So LymphoTrack is the brand name of the Invivoscribe next generation sequencing assays. These are assays for clonality testing, somatic hypermutation, as well as residual disease testing. All of the assays do follow the same 7-step procedure. They do still use multiplex PCR, so they are very similar to the gel and electrophoresis process. They have higher sensitivity, though, than those results, and they are much easier to interpret. And it is much easier to add on somatic hypermutation and MRD testing. Now, the kits that are available in India are the LymphoTrack RUO product line. This contains assays targeting the IGHV leader region for somatic hypermutation, which can also be done with the IGH Framework 1 assay. And for B-cell clonality, we have assays for IGH Frameworks 1, 2, and 3, as well as a combined assay for all of them together. We also have IG kappa for B cells. And then for T cells, we have T cell receptor gamma and T cell receptor beta. So really focused in on the two primary targets for each of the different B cell and T cell types. They're available on Illumina platforms in 8-index format for lower throughput labs and in 24-index format for higher throughput labs. And they're also available in 12-index format for ion torrent users, though we do have a slightly limited selection of targets for ion torrent platforms. Now, for LymphoTrack, you do have to plan your experiment a little bit more carefully. I did mention that the cost per sample is heavily decided by the number of samples that you will test per batch. So as you're considering LymphoTrack, you really have to think about how many samples you'll be testing, how many different gene targets you will combine together, and which of those targets you will test. And that helps to determine the number of indexes you would need, whether it's the 8 or the 12 or the 24 index set, which chemistry you might want to use, and which of the sub-chemistries, for example, XLEAP on Illumina, long read sequencing on MiSeq, or short read sequencing on NextSeq, for example. And all of that really comes down to the most important part of this, which is what is your cost per target and per sample. And as this graph will tell you, your cost per sample will always decrease as you increase the amount of targets and increase the amount of samples that you test. Here's from a publication on multiple myeloma from Sloan Kettering Cancer Center, where they show the detection rate of LymphoTrack with Framework 1, 2, and 3 individually, as well as immunoglobulin kappa. And you can see that if you were to combine Frameworks 1, 2, and 3 all together, you can get 98% detection combining these 3 targets. But most labs will actually choose to do 2 targets because this is a good compromise for high sensitivity detection without sacrificing too much cost. And you can get 97% detection using just Framework 1 and IG kappa together, and that will get you 97% detection. Of course, the ideal scenario is to test all 4 targets, but most labs have to make that calculation to decide which targets they want to test. We do try to make this as easy as possible, so you only need 20,000 reads per sample for our next generation sequencing assays. You can even use the same index across multiple different targets. So for example, if you were to have index 1, or let's say index 3 here, being tested for Framework 1 and Framework 2 and Framework 3, as well as IG kappa, you can actually use the same index for all 4 B cell targets. And you could even use the same index for a different sample as long as it's being tested for a different target. So you can see here even though we've only used 4 different indexes, we've actually tested a total of 5 samples, as well as our positive and our negative controls. So the ability to overlap the samples it really helps to cut down on that cost per test. Now, the LymphoTrack assays, they are traditionally designed for clonality assessment. We will talk about somatic hypermutation as well as residual disease testing shortly, but how is that data interpretation done for clonality detection? Now, the first thing to do is to make sure that you have enough reads. We usually suggest about 20,000 reads per sample, or at least per target, for clonality detection. We also want to check that our sequencing quality is good enough to have reliable results. And then we take a look at our top 10 merged read summary. This table has a lot of information on it, but it's quite simple to look at. This is the most frequently identified VDJ rearrangements in our sample that's tested here for IGHV leader. So number 1 is the most commonly detected sequence in this sample. Number 2 is the second most common. Number 3 is the third most common, and so on. We can see that DNA sequence here, and we can see that rank 1, for example, is 455 base pairs long. It had 50,000 reads attached to it. The V gene is identified, the J gene is identified, and the most important value here is our percentage of total reads. So the rank 1 sequence, as it represents 50,000 reads that we counted, it represents about 10.5% of total reads of the 470,000 total reads here. Now, that top rank sequence should represent a significant portion of the reads. Because these assays are RUO, we don't have clinical interpretation guidelines listed here in this presentation. However, I'll share some publications with you that show how that interpretation is done at different institutions. The final criteria is that the top rank sequence should be significantly higher than the third rank sequence. Now, that's because the third rank sequence generally represents the polyclonal background, and we want to make sure that when we have a clonal cell population, it is not just highly abundant, but it's also highly abundant over a background. Because as individuals age, if they have a potential immune issue happening, they can also have very highly elevated rearrangements in general, and that's not the same as having a true clonal rearrangement. So here we can see 10.58% is significantly a high portion here, and it is significantly higher than the 0.04% from rank 3. Now, this is, of course, the IGHV leader positive control, so this is clearly a positive sample here. We can also move in to look at the somatic hypermutation results in the same sample. Once the sample is clonal, we just take a look at the productivity of the rearrangement. Y plus Y for these two values means our clonal rearrangement is productive. So this means it can create a functional protein. And then we take a look at the somatic hypermutation rate. Most guidelines now, most laboratories, follow the European Research Initiative in CLL, or ERIC, guidelines for evaluation of somatic hypermutation, which states that the rearrangement sequence should be at least greater than 2% mutated when compared to the germline. And that would indicate a mutated IGHV. Mutated IGHV usually leads to better prognosis for the patient. Being under 2% would mean unmutated IGHV or poor prognosis for the patient. This analysis here in this example is done with the leader assay, but it can also be done with IGH framework 1. Now, just to share a publication with you, this is the major publication from the Memorial Sloan Kettering Cancer Center in New York City. They tested over 1,000 samples over 1.5 years, and showed the results for their characterization samples as well as their validation sample set. And they compared all of their results to capillary electrophoresis. And what they found was that they had 96% concordance to the electrophoresis platform. But an NGS also showed superior performance in classifying positive samples. They published a really good set of validation for how to do interpretation, DNA input, limit of detection, and the precision across different technicians. And what they found was that in dark blue here, depending on the disease type, IGH Framework 1 could detect either about 65% to up to 95% with just IGH Framework 1 of cases of these diseases here. But if you add on IGHV leader, so just Framework 1 and leader, your detection rates go up to about 88 to almost 100% of rearrangements, varying by disease type. And you can add on more targets, but of course there is a slightly diminished return as you get closer and closer to 100%. Adding on more and more targets gives you less and less return as far as detection rate. This is a publication from Europe showing interpretation criteria that they developed for T-cell receptor gamma. So this lab showed 92% sensitivity using just gamma, no beta in this case. And interestingly enough, they actually had better results for FFPE samples using NGS than they did with electrophoresis. And they developed a set of interpretation criteria that shows more than just positive negative. They broke it down to monoclonal, oligoclonal, polyclonal with minor clonal rearrangements, or polyclonal. So they showed using the LymphoTrack data, they were able to get a little bit more distinction between the different types of samples. Now moving into residual disease testing, as I mentioned, the LymphoTrack assays have been designed for clonality assessment. You can very easily take that clonality data for IGHV and IGH Framework 1 and use it for somatic hypermutation analysis. But let's talk a little bit about minimal residual disease. Now, when I showed you earlier a set of cells that was polyclonal, you could see that there was all different colors and they were equally well represented. But we need to start thinking about residual disease detection in the form of post-treatment, potentially in remission samples, where the sample appears to be diverse, it looks negative, it looks polyclonal, but if we take a look much closer, what we'll find is that there may be still that fingerprint of B cell Y being present. And there's a lot of data coming out about residual disease and its correlation with patient outcomes in the long term. How the LymphoTrack assay adapts residual disease detection, we use the same assay and the same workflow as LymphoTrack clonality testing. We simply add a few different changes to make that detection easier. So the first thing is we have a completely brand new software that has an experiment planner tool to help you get the level of sensitivity that you want to achieve. In the laboratory, there's only one wet lab change, which is to add a new LymphoQuant internal control. This is our cell control that helps to calibrate our residual disease results. And then we just adjust our amplicon, our bead cleanup volume because of the new PCR reaction volume. So very minimal laboratory changes. Primarily, your changes are going to be in the experiment planner tool in the MRD software, as well as the analysis in the new MRD software as well. There are varying needs for residual disease testing. Most institutions are interested in that conversion from 10 to the -4, which they are able to do quickly, cheaply, and easily using flow cytometry. But they really want to get down to 10 to the -5 Flow is, of course, also pushing those boundaries as well, but it's not as easy to evaluate those flow results and standardize them as it is with molecular testing like NGS. So 10 to the minus 5 is the most commonly tested sensitivity for LymphoTrack MRD assays. It requires 2,000 nanograms of DNA, which is a relatively easily achievable amount, and it requires 4.4 million reads. So you'll see that that amount of read depth has increased quite significantly. We only needed 20,000 to be able to pick out the clonal rearrangement from the polyclonal background, but now we are looking at that polyclonal population in a potentially negative sample and trying to pick that sample out that we know it's clonal rearrangement. 10 to the minus 6 is, of course, possible with the LymphoTrack MRD platform. However, we find that 20 micrograms of DNA is very difficult to achieve for most samples, and 44 million read depth is quite restrictive, at least in modern sequencing costs. So we've created the LymphoTrack bundled solution. The bundle is quite straightforward. The primary reagent that you'll be using is the same LymphoTrack assay that was used for detection of the clonal rearrangement. And what we've added is the B and T cell controls. So we have a low positive control that's 10 to the minus 4, and we've added the LymphoQuant internal control that can help to spike into your sample and help calibrate how many cells did we detect so that we can track the level of those cells over time. And the last part of the bundle is the LymphoTrack MRD software. I mentioned this has the project planner tool as well as the analysis that can be done for MRD. These are the outputs of the the LymphoTrack MRD software. You can see we create simple PDF reports to help identify low clonal cell populations. You can also track up to 5 different clonal rearrangements at the same time, and you can see that we can have a graph to watch that progression change over time. So depending on your needs for how many samples you want to track, the MRD software makes it very easy to clearly detect those on the same page or separate those out. And we can also calculate cell equivalents. And this is something new. As you saw during the interpretation for clonality results, we very frequently look at the percentage of reads that are represented by our cell of interest. But now, using the LymphoQuant internal spike-in control, we can calibrate back to the number of cells that we're detecting instead of the percentage of reads. And that's very helpful to really create these graphs and watch the level of disease go up or down. So MRD evaluation is a more new concept, or not a new concept, but a newly making it into the clinical space. This is, again, Memorial Sloan Kettering Cancer Center in New York City. This is a direct comparison of LymphoTrack to flow cytometry using 438 different time points from 251 patients. And what they found was there's a high concordance between the two methodologies. Here you can see in blue are the flow positive, NGS positive samples that are concordant. In red are the flow negative, NGS negative concordant samples. But in green is flow positive, NGS negative, and in purple are the flow negative, NGS positive samples. So, of course, these 13 samples will be of most interest to institutions that are looking to do the comparison between the two platforms. But the important part to note here is that we have about 93% concordance across the two different platforms at 10 to the minus 5 sensitivity. This publication actually shows a different story. It shows actually that NGS had better performance than flow cytometry in this lab. It was 122 BALL samples from 30 different patients. They were able to detect MRD using NGS up to 25 weeks prior to the patient's relapse. And they give a really great visual here where they had their real-time results of flow cytometry here in black. And you can see that in most cases, the results were negative, negative, negative until the patient relapsed when it comes to flow cytometry. However, when they went back and did a retrospective study in green, taking the time points and sequencing them with NGS, what they found was that they could actually detect that residual disease even when flow cytometry had been negative. And you can see they've highlighted here in this red box, when pointed at it with arrows, cases where NGS detection was positive for MRD, but flow cytometry was negative, and cases where this could have helped to predict relapse earlier. And lastly, I want to show a really interesting research application, which is repertoire analysis. So these are European astronauts that were sent to space, and they had their blood drawn before leaving, as well as on the International Space Station and when they returned. And what they found was doing a very complex repertoire analysis with the full raw data that's available with the LymphoTrack platform. They found, number one, that astronauts have more immune repertoire diversity than your average control or healthy donor. And then, of course, before the astronauts had a higher repertoire diversity amongst themselves after they had made it onto the ISS, and that higher diversity continued when they returned back to Earth, probably showing that there is a stress response that leads to a measurable change in the immune system for these astronauts. So potentially really interesting research for space travel and molecular biology mixing together in a really interesting way. And then we just have some news to share. This is really interesting and exciting information about MRD. As I mentioned, it's really a newer, more research application, but we're seeing it make it into the clinical space. In Korea, IGH and IGK assessment with LymphoTrack is reimbursed. So this testing for MRD patients, or sorry, it's MRD testing for patients is reimbursed by the national healthcare system. In Australia, there was just an approval for 1,500 Australian dollars to have MRD testing by NGS for BALL samples. And this is now also going to be reimbursed by the national healthcare system. In the United States, we had a recommendation from the committee to the FDA that MRD testing is advanced enough now that it should be allowed in clinical trials and allow pharmaceutical companies to get drug approvals faster so that treatments can make it out to patients quicker, using residual disease as a surrogate endpoint for these clinical trials. So a lot of exciting news as the development continues. That is the end of my presentation. So I really want to thank you for your time and your attention here. I'm happy to answer any questions, and I would really also like to take the opportunity to thank DSS Imagetech for organizing this webinar.
Thank you, Parth. Thank you very much, Parth, for such a informative and insightful presentation regarding Invivoscribe products as well as the technology. So now, as Parth said, we are open for the questions and answer sessions. So either you may drop it in the chat box, or you may also unmute yourself and ask the question. Okay, Parth. So basically, I have a question.
Can I ask one question? This is Dr. Srijesh. Hello. Can you hear me? Yeah. So I have a question regarding MRD testing. So if I understand, you are supplying this kit as a bundle, right? suppose I want to do MRD for BLL, I have to buy this bundle which consists of all these FR1, 2, 3 variable. Like as a cost cutting, I am talking in terms of affordability. So in the one of the previous tables, I saw that FR1, if I just test FR1, I can detect clonality in nearly 97% of BALLs. So how to go about, like, I just want to buy FR1, but I want to do MRD testing. Is it possible to do like that? Because does the beads and the software, uh, will it come along with this if I just buy the FR1 kit?
Yes, thank you for that question. It's a good question. So the results you mentioned that show the detection across the different platforms, those are for baseline clonality detection, or you can call it screening for that baseline sample. For MRD testing, we know that you need 4.4 million reads per result. So not just the cost from the assay, but even the cost from sequencing would become very high. We recommend actually you only pick one target, the target that was positive for screening. For example, sometimes if you test framework 1, 2, 3 all together for a BALL sample, you might have a positive result for all three. So you should just pick one of the three to use. Ideally, in that case, we prefer framework 1 to use for MRD testing. So for MRD, you only need one target, because if you know that target can amplify and detect your clone, then that target is a good candidate to amplify that, even when that clone is at a very low level. The bundle that we mentioned earlier is the bundle, and it's not sold as a bundle. You can actually buy them individually. We consider the solution as a bundle, which is the controls and the software that go along to make the assay. Essentially, it's a conversion from clonality detection to MRD detection, and it's just really the controls. The software is provided, so it's just adding on the low positive control, because you don't want to do MRD detection with a high positive control. It doesn't always make sense. And the internal control, which is the spike in, that will tell you, instead of saying we detected 0.001% of reads as the clonal sequence, instead it will tell you we detected 100 reads, and then it went down to 50, and then it went down to 10, and the latest time point went up to 1,000 cells. So that internal control is helpful for calibration, and those four parts together are what we call the bundle: the single assay that you select as your assay for testing, the low positive control, the internal control, and the MRD software.
Okay, thank you.
One more question has come from.
So Parth, basically there's a question in the chat box. So the question is, how about the stability of the baseline clone?
Yeah, we find that the rearrangement sequence is quite stable for most rearrangements. The challenge actually ends up being for some of those less mature malignancies. We can sometimes have a little bit of difficulty identifying the clonal rearrangement if it's unproductive, or there is background subclones that are present. But for more mature diseases like plasma cell neoplasms, myeloma, CLL, the rearrangement is usually quite stable in the clonal cell population between baseline detection and relapse, or more specifically in residual disease testing, between baseline and relapse, that MRD detection should be stable. But the nice thing is, for LymphoTrack, because you are still running the LymphoTrack assay, if a new clone is to emerge and it has a new clonal rearrangement, you will still detect it, because we're not just targeting the amplification at the MRD time point. We don't just target our clone of interest. We still continue to do multiplex PCR. We still amplify all of our polyclonal background cells. So if a new clone does emerge, you will detect that, you will see it, and it will look more like a diagnostic clone.
Okay, I hope that answered Karthik's questions. Okay, Parth. So basically, I have a question. It's just a small question. So on the MRD evaluation, where you had put on the comparison between flow cell and NGS, so where flow cell samples were being positive and the NGS samples were like in the test it was shown negative. So in that part, like how would one go about confirming that are they exactly actually positive or negative? So how would further the clinician, the
Yeah,
doctor?
so you would
Yes,
hopefully
that's all.
have a study very similar to what was done here, right? Where they had the eventual relapse cases, or hopefully the eventual negative cases as well. In this example set, a majority of those should be concordant, right? And these patients will continue to be tracked. We anticipate this institution will do another publication in the next 2 to 3 years as more data is available for all 251 patients. We want to see the flow positive, NGS positive. What are the outcomes for those patients? The flow negative, NGS negative, all these concordant ones. It will be interesting to see those results. But of course, as far as technology and us being a molecular diagnostics company, we are very interested in also seeing these 13 samples here. And what we would want to see is what are the patient outcomes for those samples. At this point, they're just considered time points and samples. But at the end of the day, we want to see something done like this, where for those 13, were there cases and time points where one or the other technology was negative or one was positive? And to be honest, I think the most likely result is that you should use both of them together. That will be the likely conclusion is use both technologies, at least until there's a one clear advantage. But right now, I think in the next 5 years, we will see concurrent use of the two platforms.
Thank you, Parth.
No other questions, Sunita?
Pardon, Suresh sir?
No questions in the chat box?
We're just waiting for a few more minutes, 1 or 2 minutes. So if we have any question dropped in the chat box, so we'll have it addressed or else we'll wrap the session.
Okay.
I received a message directly in the chat, which was about the recording. And I believe, Sunita, the recording will be available after. Is that correct?
Yes, Parth.
Great. So all registrants should get a link to the recording afterwards.
One more
Yes,
question has come.
we'll make the recording available to all the registrants after
Sunita,
the
there are two
recording.
questions available.
Okay. So are you able to read the question, Parth, or should I
Yes.
read it
I
out for
can
you?
read this. Yeah. So there's a question that says, any comparison data of LymphoTrack assay between Illumina and S5? Yes, we do. We did compare the 5 targets that are available for S5 to the LymphoTrack assays on Ion Torrent. They use similar primer design, so the detection rate is quite similar. The only difference really between the two sequencing platforms is that IGHV leader and T cell receptor beta are not available on Ion Torrent platforms. So the detection is a little bit better on Illumina systems just because you have one more target for both B cell and T cell malignancies. And there's another question, do we need to procure the software for SHM detection? You do not. There is a LymphoTrack software that does the clonality detection and somatic hypermutation detection all in one step. So those are both systems are available within the same software and they happen at the same time. So as long as you are running IGHV Leader or IGH Framework 1, you will see the MRD or the somatic hypermutation results in your top 10 read summary table. For each of the rearrangements, you'll see the mutation rate and the productivity of that sequence. Okay, there's a question about oligoclonal plasma cells that emerge during treatment. Yeah, so we do see that, especially as we start looking at MRD testing of samples that are recently post-treatment, especially with, for example, CAR T therapies, that the population of B cells actually goes quite down, and then the repertoire becomes much more restricted in later steps. Now, those samples, the real challenge with them is actually amplification. As you know, for sequencing, we have to have a certain amount of DNA before we can load it onto the sequencer. And using a multiplex PCR, if there's very little polyclonal background or clonal background, if there's very few lymphoid cells present, then we have difficulty amplifying any rearrangements to detect. So you get a very low read count, but you can still test those for the presence of your of the clone that you're tracking. Same with the oligoclonal plasma cells that emerge during treatment. If they have a different rearrangement sequence, they will be detectable, they will amplify, but they will be considered background sequences. So they might be present at 3% or 4%. But if you have an oligoclonal cell population, your rank 1 sequence, let's say it's 5%, and your rank 2 sequence is 4%, and your rank 3 sequence is 3, 2, 1, and so on, as these highly prevalent samples. Remember that for interpretation of clonality, I can actually go back a few slides. Sorry, we'll be back quite a few slides, but remember that for the detection of clonality, one of the criteria is that the rearrangement sequence should be high, but the second criteria is that it should be higher than rank 3. And when you have that oligoclonal plasma cell population, you'll see all of these percentage total read samples be a little bit high for the most frequent sequences. So as I mentioned, maybe 8, 6, 5, 4, 3, 2. It would those rearrangements would fail that second criteria for a clonality detection, and you could maybe use one of the more advanced interpretation criteria to really call that oligoclonal. But the MRD software doesn't care about the top 10. It doesn't look at only the top 10 sequences. In fact, as you see here, there are 474,000 reads present in this sample, and the top 10 table only shows about 12% of those reads. The MRD software will actually look at all 100% of reads, and it will look for our sequence of interest, which is our clonal rearrangement from diagnosis, and it will try to find that one. Does not matter if it's present with 1,000 reads, does not matter if it's present with 100 reads. I think it is 5 reads is the cutoff that it needs to be able to detect. And as long as there's at least 5 reads, then it can call that as positive or MRD detected.
Thanks, Parth.
Yeah, thank you very much to DSS Imagetech for hosting this webinar, and thank you to all the attendees for your attention.
Thank you, Parth. Thank you, everyone. So we'll basically wrap the session now.
Yes, Surita.
Okay, great.
So we can have the recording sent across, no?
Yes, that we will share it with all the participants as well as the DSS team.
Okay.
Okay, thank you, Parth.
Thank
Once
you,
again.
Cynthia, for organizing. Thanks, Parth.
Thank you.